I will not start by thanking the organizers. That doesn't seem appropriate, but we'll go ahead. So this is work done in collaboration with my student Adhya and a close friend Mastoshi, both of whom are in the audience, and Mastoshi gave a talk earlier in the day. So I'll tell you about the physics underlying cell division and how certain asymmetries arise naturally during cytokinesis. So classic pictures of cell division that we learn in textbooks. Cell division accomplishes not just the job of segregating the genetic material, but also cleaves the mother cell into two daughter cells. And the way we understand the cleavage process is by understanding that there is a big actomyosin cable that forms in the middle of the cell at the equatorial region, and the constriction forces that are generated in the actomyosin cable are what's responsible for the cleavage process to happen. So here's the picture. There's some flows that, or some other mechanism of polar relaxation, whatever it be, essentially end up giving you an actomyosin cable. The actomyosin cable has these contractile forces because of ATP activity, and the contractile forces actually pinch the cell surface. And that's the constriction picture that we all know. So this job is essentially accomplished by the actomyosin cortex, which is beneath, just located beneath the plasma membrane. It's about half a micron thick. There's a polymeric meshwork of actin filaments, cross-linkers, myosin motors, and lots of other gooey things. The predominant thing is myosin motors, which tug on actin filaments by consuming ATP. ATP hydrolysis generates dipole forces, and this at a larger scale is thought to be responsible for many cellular processes, for instance, in flow and deformation and so on and so forth. But even at larger scales, going all the way up to tissue and organismal level, a very strong correlation is that actomyosin activity is essentially responsible for many of these morphogenetic processes. Okay, so how do we understand the flows that come up by actomyosin activity? This is something that one can actually measure. So for instance, if you have the actomyosin cortex, that's a very dynamical structure in the sense that there is continuous polymerization, depolymerization of the filaments. And so on time scales much longer than the stress relaxation time scale, which is typically also the viscoelastic relaxation time scales, roughly of the order of a few tenths of seconds, the fluid nature of the cortex will kick in, so in the sense that it will flow in response to any applied stresses there. So one can actually measure these flows. For instance, in the C. elegans zygote, in the first cell of the C. elegans embryo, you can measure these flows. You're looking at the bottom plane labeled myosin, and you can do a particle imaging velocimetry, and so therefore you can get the flow profile coming out there. And so one can write down a theory to understand how this flow comes up. And the way one understands this is that one writes down force balance. Inertia is negligible at these scales, and so you write down a balance of all the internal forces, stresses, both viscous and active, and external forces like a friction. And so there's a prediction that how the flow profile should look like if I know the active stress. So this is something that one can measure. You can measure the flow profile very, very quantitatively. You can take the myosin profile as given, again from the experiments, and with some simple ansatz for how the stress depends on the myosin, you can actually predict the flow, theoretically. And so what this does is to give you handle on many of the material properties of the cortex. For instance, the ratio of friction to viscosity, the strength of the active stresses, and so on and so forth. So the upshot here is that if you have contractility gradients, contractility and active stress will be synonymous. Contractility gradients will drive flows. But this leads to a novel class of pattern-forming systems. So if you have contractility gradients that drive flows, well, surely the flows will also redistribute the concentrations that generated the contractility gradients. So there's a coupling between contractility gradients and flows. And so therefore, this system, if you work out the math, it turns out that it can actually lead to certain novel class of patterns, which are the mechanochemical patterns here. So here the heat map represents myosin concentration from low to high, and the arrows represent the vector field from the cortical flows there. So these are self-organized patterns that come up spontaneously beyond a certain level of activity. And you should think of them as analogs, mechanochemical analogs of Turing patterns, classical Turing patterns that we know. Okay, these patterns are not just theoretical, they are very useful in real experimental systems. For instance, during the first division of the C. elegans zygote, the first division is actually an asymmetric division in the sense that the two daughter cells are not the same. You can see the asymmetry both in the size, but also in the distribution of certain cytoplasmic components. This cytoplasmic asymmetry is achieved by a certain asymmetry on the surface of the cell. The surface of the cell has these polarity proteins, pars, partitioning defective proteins. They segregate red and blue on two halves of the cell, and the interface between the red and blue is where the division happens. So this is a system where one can write down a very nice theoretical model coupling the surface-bound concentration of the pars and the myosin and coupled to their cytoplasmic exchanges. But of course, myosin and myosin gradients will obviously drive the cortical flows that I talked to you about. So I'm not showing you the equations, but the generic structure is that there is rate of change of these concentrations of the red protein, blue protein, and myosin. And that happens because there's movement by advection, diffusion, and some regulatory chemistry. So the cortical flows, of course, are driven by gradients in the myosin contractility. This system of equations and the data that we obtained is so good that one can actually compare quantitatively the mathematical results with the data that you obtain, and we can actually predict the spatiotemporal emergence of the polarity pattern in the cell. So therefore, trying to understand mechanochemical patterns in this fashion is actually useful in predicting real experimental data. Okay, so one thing I want to understand is how does surface geometry affect such patterns? So for instance, if I think about a sphere of some radius r0, I can put in the same set of equations on the surface of the sphere, and turns out you do get patterns, but the patterns are like some spots which are localized at some particular region, randomly chosen depending on the random initial conditions. So nothing surprising in that. So if you include cytoplasmic flows, and in particular treat the cytoplasm as a viscous fluid, incompressible viscous fluid, put in an appropriate coupling between the cortical flows and the cytoplasmic flows, we were able to show some time ago that beyond a certain value of the activity, you can either get spot-like patterns, which are very similar to this, which is like a cell polarity pattern, or you could get ring-like patterns, which is like the cytokinetic ring that you're thinking about. So you think about the concentration C as some analog of the actomyosin concentration field. So this mode, where the length scale, that is the ratio of the cortex shear viscosity to the cytoplasmic shear viscosity, compared with the radius of the cell, here assumed to be a sphere, sort of dictates which kind of pattern you get. So when this length scale is much larger compared to the radius, you get a spot, it's like a polar pattern. When the other way around, you get a ring-like pattern coming out spontaneously. However, if you plug in the values for the silicon zygote, this ratio turns out to be in the other direction. So you want LH by R0 to be much less than 1. However, this turns out to be, this is the worst guesstimate that we can do. And even the worst estimate shows it's of the order of 10. So therefore, it's unlikely that cytoplasmic flows are the predominant reason why you get ring-like patterns coming out. All right. The kind of patterns that I've been showing you are patterns of actin and myosin and velocity fields. But remember, these are all patterns on fixed surfaces. So I showed you things on two-dimensional domains, maybe on sphere, maybe you could do for non-spherical shapes as well, but still the geometry is fixed. The cell division is a problem where you not only have patterns on a domain, but the geometry of the domain itself is changing with time, right? And the geometry change is influenced by the patterns here, and so therefore there's a feedback. So that's something we should include. So there's not just patterns, but there's also shape, and the patterns and the shape talk to each other very, very consistently. So for physicists, this is essentially geometrodynamics. And so geometrodynamics of active surfaces is what we want to understand clearly. Okay, so how do we build this? I'll provide a very simple picture. So let's say you have some surface, and you're thinking about the surface as something sitting in three-dimensional space. You look at the normal, the unit normal to the surface at every point. The unit normal, the way the unit normal changes as you go around the surface, is essentially the surface curvature tensor. Okay. So once you have the surface curvature tensor, you can uniquely define the surface embedded in 3D up to global translations and rotations. Now, that's the geometry, but we also want to think about the flow. So in other words, we have the cortical flow V. So I'll decompose that into a part which is tangential to the surface and a part which is normal to the surface. Now, if you have a gradients in the normal flow, so one can show that that will actually change the normal vector. So therefore, the shape changes, in other words, the local tangent planes change or the unit vector changes because there are gradients in the normal component of the flow. Now, once you have these flows, the points that lie on the surface of the sphere or the surface of the cell will actually move according to the normal velocity there itself. So this is assuming I've given you a flow and you can tell me how the shape changes subsequently. But where does the flow come from? Well, that's where the active forces should come into the game. So remember this picture that I painted for you, that if you have myosin, moves around by advection, diffusion, some simple regulatory turnover. Myosin concentrations generates active stresses. Here I assume for simplicity, isotropic active stresses. This is in addition to the typical viscous and bending stresses that would be there on a deformable surface. So I do a force and torque balance. So I write down the divergence of the stresses and balance out with frictional forces and also some pressure to account for volume constraint of the surface. So this gives me a way of taking in a concentration profile and giving me a velocity profile. But then now I use that velocity profile to change the shape and move the surface. So therefore, there is this coupling that I implement between pattern and shape. I'd like to ask, what are the kinds of patterns that will come up spontaneously as you vary the activity patterns? Here's the picture. So I want to start from something which is unpatterned. So some patterns will spontaneously develop, patterns of concentration, maybe flow, maybe stresses and so on. So I'm going to use those stress patterns to deform the shape. Well, once you deform the shape, surely it's going to affect the patterns back. Did I stop? Yeah. Can you hear me? No? Yeah. So once you have the There's a bit of an echo. Hello? Yeah. Once you have these patterns, the patterns of flow and concentration will also generate patterns of stress. Now, I use those patterns of stress to deform the material. But once you deform the material, well, that'll have a feedback on the pattern because the geometry has changed now. The geometry affects the kind of patterns. So I want to see what are the kinds of emergent patterns of myosin and shape. So that's the way I try to think of the implementation between pattern and shape coming out automatically. So the main result of the talk is the following. We see cytokinetic patterns emerge spontaneously. So we start from a sphere with some uniform concentration of myosin with small random fluctuations around. I'm plotting on the right side the deformation rate, basically the normal component of the flow. And beyond a certain activity, we see the spontaneous emergence of both a ring-like pattern of myosin and the concomitant shape change of the cell surface, right? So the movie is going backward and forward so you can see the thing coming up. So there's spontaneous arrangement, there's spontaneous patterns of myosin come up, you can see a ring there, and that's the flow profile there. The arrows indicate the tangential component of the velocity. So here's some snapshots. So you start from something which is unpatterned, and you go towards a pattern region where you get a spontaneous ring with a concomitant deformation of the surface. Okay. So these patterns seem to emerge very spontaneously. So I want to show you how these patterns come up. So let's break up the viewpoint. So you can look at the transverse plane, the plane which is perpendicular to the division plane, and also the division plane, and I look at the kinds of patterns that come up here. So this is the outline of the periphery of the cell along the color coding given by the concentration of myosin. And you very clearly see that in the division plane, as the division proceeds, there is an enhanced myosin accumulation in the ring. But not only that, you also see the accumulation of the myosin in the transverse direction as well coming up very nicely. Okay, so therefore there's a phase diagram. So as I vary the material parameters, I get different kinds of patterns of shape and concentration. So what are the kinds of shapes and patterns that you get? So if you don't have any contractility or very low contractility, obviously you get something which is boring, uniform myosin and a spherical shape. So that's just the homogeneous patterns. So if you crank up the contractility at certain regime, you hit a parameters where you start getting spots, single spots. Remember the spots that I showed you on the sphere. But if you go up higher in contractility, you spontaneously start getting rings. So the ring-like patterns do not result from a linear instability of the homogeneous. There's a secondary instability of the spot-like pattern. So if you do the standard analytical linear stability calculation, you'll never predict this. So you need the full three-dimensional numerics to actually see that there are indeed ring-like patterns that are coming up. All right, this is all theory. Does it work in the real cell? To do that, Masatoshi has a fantastic imaging system where he actually labels myosin, follows it in space and time. So we do confocal microscopy, construct Z-stacks, and then reconstruct the three-dimensional image of the cell from this data. And you should see that as time goes on, you see the spontaneous formation of the ring and the subsequent constriction of the ring to give you cytokinesis happening simultaneously. So this is data accumulated from myosin intensity maps for several time points. Okay, so let's look at the chymographs. So this is theory from my theoretical calculations, Adhya, not mine, theoretical calculations, and this is time going forward. And you see the intensity of myosin coming up, and you see a formation of a ring-like structure. You see something very qualitative coming up in the experiment as well. I can also plot the ingression rate. This is the rate at which the cytokinetic ring is going inward, and you see something very qualitatively similar between the theory and the experiment. All right, so this is qualitative. We want to get quantitative comparisons as well. So one thing one could do is the following. As the ring constricts, you can measure the radius of the ring and plot the radius as a function of time. And so if you plot the radius as a function of time in the theory, you see that after some transient regime, there is a regime where the radius decreases linearly with time. So in other words, the ingression rate seems to be a constant. Okay, so U-perp is the ingression rate. This is something people have seen in many cells, not just C. elegans. There are sea urchin species and several other examples which actually show that the width of the ingression furrow goes down linearly with time. So this is something that we did not put in the theory. We just put in force balance, physics, geometry, and this result just pops out. So the conclusion is that the cytokinetic ring seems to ingress at a constant rate. But not just that, the rate at which the ingression happens seems to increase with the activity. So the Peclet is a measure, some non-dimensional measure of the amount of contractility. So higher the contractility, the faster the ingression that comes up there. Again, can we test this in experiments? Well, here's the idea. So we were thinking about this coupling between patterns and shapes. So now I'm going to switch off the pattern part and just look at how the shape evolves and do the kind of perturbations. C. elegans is nice because there's a whole plethora of perturbations that you can do. There's a library of perturbations which can increase the contractility, decrease the contractility, and whatnot. So we can look at this library, do a couple of knockouts of certain genes, and so therefore ask for the emergent phenotypes. So there's a wild type in red, and that's the one that I showed you earlier. But if you do perturbations, say GSP1 or NMY2 to 1, you do indeed perturb the constriction rate. So therefore, you see the slopes, the straight line slopes, that indeed changes with the perturbation. So if you do NMY2 knockout, obviously you're going to reduce the flows, and so therefore the slope actually goes down. So this idea that the ingression rate actually increases with activity, which came as a result of our theory, is borne out in the experiments. Okay, the other part. So one is the shape aspect, the other aspect is of the active patterns. In other words, the accumulation of myosin in the cytokinetic ring. So to do that, we integrate the myosin over the ring and look at the average amount of myosin in the ring. Remember, the ring is constricting, but you're also trying to measure the average amount of myosin in the ring. So our theory predicts that there's going to be an exponential regime, that beyond a certain point, the myosin in the ring increases exponentially with time. This is something that the theory predicts. And lo and behold, from the experiments too, we see something very, very similar. There's a log linear scale, straight line here means an exponential increase. So the accumulation of myosin is exponential, and the accumulation rate, kappa m, increases with activity. That's something, again, predicted by the theory, that the rate, the slope of these lines goes up as you increase the contractility. And that's something we also see when you do the experiments with varying the contractile flows. Okay, so I showed you two things. One is the cytokinetic ring ingresses at a constant rate, and the average myosin goes up exponentially with time. So there are these two rate constants, the ingression rate and the exponential rate. Is there some relation between the two things? This is something we found out in the theory. We asked, how does this ingression rate, the myosin accumulation rate depend on the ingression rate? And we can vary the Peclet number, basically the contractility, and vary also some other parameters like the hydrodynamic length and turnover rate and so on and so forth. We see some sort of a positive correlation between the increase in the myosin rate and the ingression rate. And this is something we see across various values of the contractility. So it seems to be some sort of a master curve that's being selected for. Similar thing we can do in the experiments, same ideas, measure the radius, measure the myosin, look at the rate at which they change, and the experiment also shows something very, very similar. Again, this is something we predicted from the theory, we just measured in the experiment and it seems to work. Okay, so the upshot is that the theory of active deformable surfaces seems to quantitatively capture the kinetics. It's just the kinetics of the ingression of both the ingression rate and the myosin accumulation in the ring, and some universal features between these two rates seem to be quantitatively captured by the theory. But there's not just that. There's something else which seems to appear very naturally in our theory. So if you look at the movie of the first cell division of C. elegans again, I pointed to you that there is the asymmetric cell division. The two daughter cells are different in size. You can see that very clearly as a difference in the size of the two daughter cells. But not just that, there's another kind of asymmetry. So if you label the membrane and you follow the subsequent ingression of the membrane, you'll notice very clearly that one side ingresses more compared to the other side. So this is not like the standard textbook picture of cell division where there's a cytokinetic ring and the ring ingresses at a uniform rate. Different regions ingress at different rates. So again, if you look at in the division plane, the cytokinetic ring that you see does not stay put at the center, but just slips. So here's the time sequence. So what you're saying is that the cytokinetic ring actually displaces away from its center. Now, this is not specific to C. elegans. Turns out asymmetric ingression is seen across all species, many, many species. Here's a fantastic movie that I lifted off Twitter when it was called Twitter. You see, this is comb jelly, and comb jelly, the ingression happens only on one side. The other side does not even ingress at all. You can see similar things coming up in the C star. Of course, there are other correlations. For instance, in this case, the spindle is localized to one side. So in fact, Rappaport's book lists a plethora of species that actually show asymmetric ingression. So naturally, the question is, why asymmetry? And is this something that can come about from the simple physics? Again, to quantify all this, you can ask questions, well, maybe there's a signal. I mean, that's the first thing we would say. Turns out that as far as we can make out, and also consistent with previous reports, the orientation of the asymmetry is random. Every time you repeat the experiment, it seems to come from a different place. It's also sort of persistent, even if you put the embryo along gravity. So, I mean, you can put the embryo along gravity, not between the glass plates, and the cross sections are much more nicer and circular, but the ingression is still asymmetric. As far as we can make out, the spindle is not asymmetrically positioned during this process in C. elegans. And all these things we have reconfirmed. So maybe to the first order, the spindle is not the reason why there is an asymmetry in the ingression part. Does this come out from our theory? So remember the plots that I showed you earlier? If you did not notice, they were all asymmetrically ingressing, but I'll just pronounce that much more clearly here. So if you have low activity, low contractility, you get division, and the division seems to be symmetric. If you look at the distribution of amount of myosin, that's more or less symmetric. However, if you increase the contractility, the distribution is asymmetrical. There's more myosin here, less myosin here. So asymmetric ingression seems to come out very naturally from our theory. Nothing else to be put in. Can we quantify that? Is this just a qualitative thing? Again, we can measure not just the radius, but also the shift in the center of the cytokinetic ring, follow that as a function of time. I just told you that if you increase the contractility, the shift becomes higher. Conversely, if you decrease the contractility, it becomes more symmetric ingression. So we can measure this asymmetry as a function of time. Again, we seem to see a regime where there is an exponential increase in this asymmetry before there is a tapering off, something that we see very, very clearly even in the experiments across RNA conditions. Okay. So the rate of increase of this slope, the K delta that I call, that itself seems to be something that increases with activity. Well, surely, just like I asked you earlier, you can ask, is there a relationship between K delta, the rate at which the asymmetry increases, and the ingression rate of the ring itself? This is something we got in the theory, again, by varying the Peclet, the contractility, and the hydrodynamic length, and the turnover rate, and there seems to be a much more nicer master curve coming up here, something that is very well captured even in the experimental data. We've done about 9 RNA perturbations, all of them seem to fall on the same master curve coming up there. So the upshot is that if you have higher the contractility, the higher the asymmetry that you get of the ingression picture. So the theory of active deformable surfaces not just captures the fact that there is cytokinetic ingression, but also the emergent asymmetries in the cytokinetic ingression. So in fact, if you look at the phase diagram, this time in the contractility and some hydrodynamic length phase diagram, hydrodynamic length plane, there's a homogeneous state, there's a spot state, and there's a ring state. So what we find is that you get most symmetric rings when you're very close to the boundary between the rings and the spots. As you increase the contractility, you get more and more asymmetric rings. So it looks like the asymmetries that you see are a very natural consequence of the physics, and there's nothing one can do. If you want symmetry, you probably need more regulation. So this raises the idea, maybe evolutionarily older creatures had asymmetric ingression, and the ones that came up later have invented mechanisms to make it symmetric, if that's required. All right, I'll summarize that. So what we've shown is that the patterns of myosin and shape that are seen in cytokinesis seem to emerge very spontaneously just from the physics, geometry, and active mechanics. Once cytokinesis is initiated, maybe has some other coupling to cell division and so on, our story is suggesting that the subsequent spatiotemporal patterns of both the stress regulators and the geometry seem to be completely self-organized. Physics is enough to drive this entire thing. And this kind of a patterning emerges because there's a coupling between the concentrations, flow, and the geometry is dynamical here. Not only that, any kind of small inhomogeneities in the myosin is amplified by the active flows, which is coupling the speed, and so therefore also gives rise to the asymmetries that you see during cytokinesis. All right, let's stop there. Adhya is the one who did the majority of the work. Ma did all the experiments, and there are people who fund me. Thank you.
All right, questions?
I'm just curious about how you increase contractility and whether there's any interesting genes that you found in the C. elegans study that were surprising or not known to influence actomyosin contractility.
So like I showed, there's a library of gene perturbations that one can do in C. elegans, which actually gives you, this is a measure of the cortical speed, which is really a direct output of the contractility.
Right.
And so there are genes, this is the wild type somewhere here. I think that's the wild type, yeah. There are genes which increase the contractility, there are genes which decrease the contractility.
Right.
So there's a map from the genotype to the phenotype. We don't understand the map, but that's something we can find out experimentally.
Okay. But I was just curious whether there was anything surprising in that group that increased contractility that you didn't know before would as an influencer of contractility.
None that we can think of. These were genes that were studied very well.
Can you ask? What happens to the region in the phase diagram where you get spots? What is the shape which you get?
So this is like a pancake kind of a thing. There's a single spot and it has deformed from a sphere to some flattish region. That's a stationary state. So nothing, no. I mean, it can show some net movement, of course, but there's no ring-like shapes that come up.
Sorry. So your initial condition for the myosin is that it's essentially randomly distributed, but it doesn't necessarily have to be there. And as you were sort of saying at the end, higher organisms tend to have more regulation. So are you able to essentially see what happens if you, for example, have myosin higher in one pole than the other, and does that have distinct behaviors?
Yeah. So Adhya has tried a bunch of initial conditions. We could start with the ring, something at the pole, not at the pole. But if you're in the regime where the rings don't form, these things just relax out. I mean, you might get some transient dynamics, but it doesn't self-organize into a ring. You need to be in the right Peclet turnover space where the rings will self-organize.
And there's a second question, and maybe I'm completely misunderstanding, but can you apply this to things like columnar epiphilia and ask how they, rather than how they start round and go asymmetric, how they go from being elongated to round in order to divide?
Yeah. So one thing that Adhya has tried is, again, not to start with a spherical shape. Let's say you make an elongated shape. You actually see a rounding up before the ring-like structure seems to form. We're not worried about confinement effects here, like in the epithelia. That will, of course, complicate matters. Even in the C. elegans, there's, of course, the eggshell, which is confining it. Sans those effects, the kinetics seems to be quite general.
Quick question. So in the mutants that increase contractility, that in which you see a higher propensity for asymmetric ingression, what happens to the heterogeneity
of what?
What happens to the heterogeneity in myosin? I mean, so I'm just wondering, are you just reaching a sort of high, is it important to reach some threshold level of myosin?
No, contractility. So remember, there'll always be spontaneous heterogeneity. There'll always be fluctuation. The question is whether those heterogeneities get amplified before they get relaxed by, say, diffusion or turnover. So that requires you to have higher contractility. Diffusion and turnover will always homogenize things. So therefore, if you have higher contractility, you can get asymmetries, inhomogeneities, which get amplified to become asymmetries.
Very impressive work. Something not directly related to the talk, but in protein aggregation, if we see ring-like oligomers are formed, which transforms to linear oligomers.
Transform to?
Linear oligomers. Yeah. Can you say something about the concept linked to linear in this?
Not directly, because remember the level where I'm building up a theoretical model is not at the level of the individual molecules, right? I'm looking at a much coarser level, looking at densities and fields like that. So what you're asking at a single molecule
no, multiple molecules. So to be more precise, let's say we have different molecules. Do you think the size, the number of molecules determine the compressibility and henceforth the geometry? So let's say if we have 6 monomers.
I see.
So 6 monomers forms ring-like oligomers. If we keep on adding the monomers, and let's say we go to about 8 like monomers or 10 or 12.
8, not of the entire surface.
Yes.
I don't know that system very well. Okay. We can discuss.
Yeah, sure. Thank you.
Right. In interest of time, let's thank Vijay for his wonderful talk, and then let's move on to